People like you and me have immune systems to help us fight off diseases, but what about bacteria? When a virus attacks a bacterium, it injects its genetic code to hijack and force the cell to make thousands of copies of itself before bursting out, which kills the cell in the process. Obviously, this outcome is not so favorable for the bacteria, which is why, just like humans, they have defense systems of their own. CRISPR-Cas9. Whenever a new virus attacks a bacterium, the bacterium cuts off a snippet of its genetic code and incorporates it as a spacer into the bacterium's DNA CRISPR array. This process is called spacer acquisition. By adding to this list of snippets, the bacterium updates its database of harmful viruses based on their genetic codes, which can be thought of as their genetic fingerprints. The bacterium then makes copies of these fingerprints in the form of guide RNAs in a process called CRISPR RNA biogenesis. These guide RNAs are then released to the rest of the cell so that it's alert and aware. The bacterium only needs to act on this released information, and this is where CRISPR-Cas9 comes into play. Whenever one released guide RNA and one manufactured Cas9 protein come together, one CRISPR-Cas9 is formed. The guide RNA, the locator, the copy of the genetic fingerprint, leads the way and scans suspicious DNA. If there's a match, then Cas9 the effector will be activated to cut it in half, disabling the invading virus. This last step is called interference and happens whenever any guide RNA verifies a virus invasion. So for decades, scientists have been trying to edit genes, but the problem was that the genetic locators they put together were either too jumbled up to be effective, often giving mixed signals, leading the genetic scissors to cut at the wrong place, or too complex to constantly recreate with so many individual pieces to put together, making it time-consuming and costly to build. That's why when guide RNA and Cas9 were discovered, it was revolutionary. The bacteria's natural defense mechanism CRISPR-Cas9 could be effectively adapted for scientific research. With guide RNA's simplicity being only one continuous genetic strand, the locator can now be made much more easily. With guide RNA's effective programmability, any genetic address could be programmed into the locator and the effector can now be accurately sent there to make edits. So as you can see, it's a premium package right from nature. But won't Cas9's genetic scissors just destroy the DNA, like what happened to the virus? Well, scientists eventually realized that if they simply deactivated the two scissors domains, the H&H and the RUVC, and added on different beneficial accessory proteins, instead of destroying the DNA upon arrival, modified Cas9 could actually make different types of beneficial edits. Therefore, because of these advantageous properties, the programmability of guide RNA, the tailorability of Cas9, the pre-existing compatibility between both, CRISPR-Cas9 is a very efficient, effective, and convenient tool that can be used to diagnose diseases, cure diseases, and possibly even treat cancer. Just as how humans learned about flight from birds and thus invented new ways of transportation, so too did they learn about CRISPR-Cas9 from bacteria and thus invent new ways to save lives. By taking time to understand the forces of the natural world, we are able to learn from them and eventually harness their powers for the good. My name is Susanna, and thanks for watching.